Welcome to the translational symposium for the fall meeting. For those of you who I haven't met yet, I'm along with Dave Tuvison, who's sitting back there next to Larry. We're from Cold Spring Harbor Laboratory, probably the newest member of SWOG, and we're very happy to be here. Larry asked me to do this translational symposium last spring and to try it again this fall. And the idea was to bring the forefront of genomics and genetics up against the realities of clinical medicine and see where the boundaries lie and how things will proceed. So the subject we picked was circulating tumor cells in cancer research. And the reason I picked that, of course, is because that's what I work on. But it's also because, is it working? Oh, because it's a hot topic right now. And the speakers tonight, this afternoon, will really represent well the different directions that this is going. But it's really hot, as you can see, because if you just look on the internet, it runs off the bottom of the slide, but these are the companies that are listed as having efforts in circulating tumor cells. So there's probably 50 labs and 50 companies all vying for being the key to really understanding and making circulating tumor cells into a useful assay. But the only one that's actually gone the distance and had it approved is, of course, the Veridex cell search assay. And that's really been a huge, it's been a huge effort for them, but also a huge boost for being able to actually have an FDA-approved assay that we can employ in clinical trials. And they, through means that I think Dan Hayes will talk about, actually isolate circulating tumor cells through a ferromagnetic fluid, and observe them. But as you can see from one of the slides that in their package, what we're looking at in circulating tumor cells is a pretty big mess. There's lots of different forms. There's lots to understand. And so Dan will probably focus a lot on the Veridex technology and how that's being used currently. As I said, they've made a huge leap forward, but every lab and company is following after Veridex is modifying capture technologies and trying to and claiming larger and larger numbers of circulating tumor cells being captured. I think that there's a great room for improvement. However, not all of the things that they capture may be relevant, and we'll have some comment on that, I'm sure. All of these various technologies and various big boxes are all being developed just to make circulating tumor cell assays more efficient in one hand, but also making the cells available to not just observe, but treat biochemically and through molecular biology techniques. And that's the point of my introduction, is that beyond counting cells, we have to make them available in a form where we can actually look at the RNA, DNA, and proteins in the most advanced ways. Howard Shear will talk about who's obviously well known for all of his clinical trial work, especially most recently in getting the Medivation drug approved for castrate-resistant prostate cancer. However, big an improvement this is in getting the drug approved, what you'd really like is to see a survival line, at least for some patients, that was 100% reduction in risk of death. And that's very difficult in a case where you've got many multiple metastases. all of which could be evolving separately. And the only logical way to look at that is through a fluid biopsy where circulating cells could represent all of the different metastases, and we'd be able to follow the ones that are being treated and the ones that are becoming resistant. Stephanie Jeffrey with the Stanford Bioengineering Group has taken several different strategies, one of which is very, very clever with Stephen Quake, the mag sweeper technology that she, I hope we'll talk about some. She's the first person to be able to trap single cells and look at the RNA profiles. Here she is, looks like she's trying to trap them by hand or else build the machine. And so my point on the introduction is that we're at an interesting point now. There are genomic techniques being applied to circulating tumor cells, but there's a huge amount of work to do, and we have to separate two things. One is the discovery, which is going to involve a lot of work, a lot of cells, a lot of cases, and you can't deep sequence every circulating tumor cell in every case. So we've got to find ways to make that more efficient. We'll talk about one that Howard Shearer and I collaborate on. But that's different from what things will eventually be when they're actually approved diagnostics, where if we use the discovery approach, we'll be able to focus on specific markers, and that's the real endpoint where we can develop approvable assays that are focused on specific genes or specific markers. And so to start off, I wanted to introduce Dan Hayes since I had pictures of everyone else. When I went on the internet, it turns out Dan has a very complicated and very varied life. I first found dominated by his days in jazz entertainment, and then, of course, his time as a stand-up comic where he was interviewed by Jay Leno. And then this version of Dan Hayes, who pitched for the San Diego Padres. But then we were passing up this version of Dan Hayes. Everyone has bad days. But finally, I found the real thing. Dr. Dan Hayes, as you all know, a doyen of clinical trials and of breast cancer medicine at Michigan